Okay, so I'd like to firstly express my gratitude to the organising committee for giving me the opportunity to come talk to you today and share some unpublished data of one of the studies I've been carrying out into BCR signalling in CLL. So it's already been put very eloquently that it's still an amazing thing that we're asking biological questions of a pathway which, relatively speaking, is well annotated, but of course we are. And some of these include the nature of the response to antigen, what influence different therapies have, and how the BCR signaling response evolves throughout a patient's disease course. And though genetic and epigenetic studies have begun to investigate aspects of this, the insight that they provide is offset by the lack of a protein-based approach, which has the resolution to complement their findings. And so the work that I do tries to reconcile these two together, and what I'd like to share with you is one of the protein-based approaches that I've developed for investigating the BCR response in CLL, and this involves the use of kinase. So these are immobilized kinase inhibitors, which are able to, or have the capacity to isolate large numbers of kinases from whole cell lysates, which are recognized through mass spectrometry. And therefore, this acts as a very appealing method, but this is not just in terms of protein expression, due to the interaction with kinases being related to the availability of a kinase's active site, and this has been meant that it's possible to determine change in activation status in relation to different conditions, and this has been shown most prominently through the work of Gary Johnson and Bernard Kuster, who've shown that in the presence of stimuli, if cells are able to respond, then there's an increased level of binding of kinases to kinases, so it's possible to determine differences between indolent and responsive signaling. Comparatively, if a kinase inhibitor is present, then this interferes with binding of kinases to the kinases, and therefore it's possible to distinguish disrupted signaling. So I've adopted and optimized this to investigate the BCR response in primary CLL cells, and I'm going to share with you data specifically for activation of surface IgM involving beads conjugated to a pan-protein kinase inhibitor called Kinet, or CTX. So this cartoon just gives an overview of how this has been applied to a cohort of almost 50 patients, which compares their surface IgM activation compared to baseline for 5 minutes. And lysates from these conditions are incorporated into the assay, and following interaction with kinases can be eluted and processed before being tested via mass spec. Max Quant software is used to determine kinase identity and their intensity, and through a simple fold change ratio comparison of stimulation versus baseline, it's possible to determine activation change in kinases in response to stimulation, and through that method it's been possible to identify a signature or fingerprint, if you like, of over 100 kinases, which demonstrate change across the entire cohort. These can be assembled into heat maps, so within these, each column represents an individual patient stratified across that fingerprint, and with the red meaning increased signaling, the blue meaning more indolent signaling. And what immediately stands out is just how varied each patient's response is, and that's largely one of the key findings, that everybody or every single patient demonstrates a unique kinome response following stimulation. And when these have been stratified accordingly based on different signaling and prognostic indicators, though patterns emerge, there's nothing really that's clear cut. So when this data was scrutinized a bit further, it was found it's better, or we found better definition or better differentiation based on therapy, because we found that patients who had previously been treated with using chemoimmunotherapy demonstrated more intensive signaling compared to patients who were treatment naive. So this gave us the first suggestion, if you like, that patients undergoing therapy could be having their signaling perturbed, and reprogramming the signaling in relation to therapy is a notion which has been put forward in tumours before, but it was intriguing that it could be taking place in the BCR pathway. So the next stage was to try and investigate this in a bit more in depth and use matched sample analysis. And to do that, we've turned to clinical trial samples. So continuing along the theme of chemoimmunotherapy, we did paired sample analysis of 10 patients who'd been recruited to Arctic and MI, comparing their baseline, so pre-trial therapy sample, versus activation of the sample taken at disease progression. And what was noticeable is that the unique nature of these kinome responses can be found not only between patients, but within patients, showing how unique they are at different time points. Not only that, it's possible to distinguish how very indolent signaling in some patients becomes far more intensive at key time points within the disease. This is not just in the case of this type of therapy either, because we've started to see it in terms of ibrutinib. So we've tested patients who are attending our clinics as well as those recruited to ISCOR. And then which largely this shows the pilot study we did before patients recruited to that trial, comparing their pre-ibrutinib samples and then 1 and 6 months following initiation of ibrutinib. And what we found was that in 3 of these, you see this far more intensive signaling at 1 month following treatment, and that's in spite of the loss of BTK, and we can validate this. So this is immunoblotting of kind B dilutions taken from 2 representative patients, comparing their baseline sample in 1 month following initiation of ibrutinib, and what you can see is there is a loss of BTK following the in vivo action of the drug. Interestingly, at 6 months, we start to see resolution, if you like, of the signaling. However, it's also possible to identify patients whose signaling persists. So this is a notion that we're trying to propose, that therapy and time are major influences on how the CLL cells respond to therapy or respond to stimulation, and how therapy could change that. So more recent work has been trying to find key drivers that promote this, because 104 kinases is still a lot to wade through. And so for that, we've recruited the help of our colleagues in the clinical trials unit, more specifically for biostats analysis. And through their exploratory analysis, it's been possible to refine the fingerprint from 104 down to 12 kinase fingerprints. Network analysis has shown that 10 of these 12 kinases directly interact with each other on an experimental level. So we have a signaling nexus, if you like, which we can use to interrogate our other data set. And when we've done that, we found that previously treated patients demonstrate greater levels of activation change in certain kinases. And some examples here are shown. I don't need to introduce SIK and FACT2, but some other interesting examples include GAC and RISC2, both of which promote proliferation. In the case of RISC2, also promotes cell survival. So there's this implication, if you will, that changes to the signaling wrought by therapy could also be having a knock-on effect potentially to the biology of the cell as well. So I'd like to quickly summarize. So I'm hoping that I convince you of the existence of a method which can be used to profile signaling changes to a high extent on a protein level. And this method is able to show that response to surface IgM activation leads to a unique kinase response or kinome response, which also may be more widespread than we may already think, because this, if you like, is a reimagining of the Cancer Atlas, which was published in Cell a number of years ago, just reimagined for kinome of pre-treated patients, just showing the number of kinases which we can detect and just the extent to which they change, and the sheer number of pathways that appear to be altered in response to surface IgM activation. And we propose that we have some evidence now that therapy has the potential to perturb or reprogram the signaling response of CLL cells, and that may have implications for the biology of the cells as well, but also provide potential new nodes for us to investigate. So I'd like to finish by firstly express my gratitude to all the patients who have donated cells for these studies, all my colleagues who work in Liverpool, the biobanks, both the domestic and the trials biobank, as well as the clinical trials team, both at Liverpool as well as Birmingham and Leeds, for all their help and support, my collaborators in Warwick and my former group in Southampton for all their mentorship and continued collaboration, and my gratitude to all these funders who provide the basis for the work. Thank you. I'm more than happy to answer questions.
Okay, the paper is now open for discussion. Are there any questions? Okay, perhaps while we're waiting for some people to think of some questions, you've obviously looked at anti-IgM signaling, and we've talked earlier about how ANAG perhaps is reversible. Do we know, one, how reproducible this assay is over time if you take the cells at different times within the same sample,
yes,
untreated?
Yes, so I mean, we've talked about this at length, but yes, that's been done over time courses. So we've done, if you like, combined analysis comparing recovery, say, of IgM expression over time, and then what that means to the kinome expression. And we compare the baseline, so the unstimulated as well as that. So we do actually start to see recovery of that and some stronger responses, but I can't really go into much detail about that because that's hopefully going to be published. But we have done it, yes.
And perhaps second quick question, do we know how IgD, does it differ from M? Are you able to say anything on that?
So I can say, because we have done it extensively. We do see actually remarkable similarity between M and D, but we've begun to notice some key differences that exist. And so that's something that we want to explore, is whether or not where the divergence is and how that could be exploited as well.
Brilliant. Okay then, well, thank you very much, Adam. Thank you.